It's lovely to see so many people from different parts of the world. As everybody sort of gets settled into the Zoom, one thing I will let you know is that one of our speakers is not able to attend today. So Professor Bhatia is kindly agreed to go ahead with the conversation today, and will present the biological overview of ASH, but Professor Mike Morrow is not able to join us. So what we're going to do is we're going to record the clinical highlights of ASH at a later date, and that will be available online. Again, welcome to our last iCMLf clinical conversation for the 2022. It's been an interesting year as we go through getting used to sort of meeting people in person while still doing these online webinars. So as this last one, we are doing a follow-up from the recent American Society of Hematology meeting. And as I mentioned, Professor Ravi Bhatia from the University of Alabama will be doing the biological overview, his perspective on the highlights. But Professor Michael Morrow, who was due to do the clinical overview, will not be joining us today, and his recording will be available at a later date. So without further ado, I'm going to introduce Professor Ravi Bhatia, who I know that many of you know. So Professor Ravi Bhatia is Director of the Stem Cell Program at the University of Alabama in Birmingham, USA. From 2015 to 2021, he was also the Director of Division of Hematology and Oncology and the Deputy Director of the O'Neill Comprehensive Cancer Center. Prior to joining the University of Alabama, Ravi Bhatia was Director of the Division of Hematopoietic Stem Cell and Leukemia Research and Co-Director of the Hematological Malignancies Program in the City of Hope Comprehensive Cancer Center. In addition to his extensive experience in studying normal and malignant hematopoietic stem cell growth and therapeutic targeting of leukemia stem cells in the translational laboratory setting, Ravi is also a practicing clinical hematologist-oncologist and hematopoietic cell transplant physician. So as I mentioned, today Professor Bhatia will share with us the key biological highlights for CML from the recent ASH meeting, and we are very privileged to have him with us today.
So thank you very much, Nicola, for that kind introduction. It's really my pleasure and privilege to be presenting today. I'm sorry that Mike can't be here with us, so we're missing an important aspect of this conversation. But I'll go ahead and present an overview of the abstracts related to biology that were presented at ASH 2022. Thank you all for being here today. It was really nice to be able to get together and attend ASH in person after 3 years of the pandemic, where the meeting was largely virtual. So I'm going to cover a few different topics from the biological perspective. The first is going to be related to pretreatment biological characteristics that determine response in CML. The second is going to be on novel approaches to targeting leukemia stem cells. And finally, I will be talking about metabolic vulnerabilities in leukemia stem cells. So this was a very important presentation from Singapore, from Duke and US, which was presented by Vaidehi Krishnan and from the lab of Sin Tiong Ong. And this was titled, "A Single Cell Atlas Identifies Pretreatment Features of Primary Imatinib Resistance in CML." And the premise of this study was that we know that TKI, there's heterogeneity in TKI response. And this they classified into 3 broad groups. Good response, so group A were patients who achieved an MMR by 1 year and long-term achieved a deep molecular response. And group C, which was the failure group, had basically TKI failure and blast crisis progression. And then there was an intermediate group with an inferior response, group B, which did not achieve cytogenetic or molecular response and required TKI switching. And the question that they asked was that if they did single cell RNA sequencing at the time of diagnosis, could they identify factors that predicted subsequent response? And so they took patients from the three groups, which was basically, I think, covered a number of the patients that were treated at Singapore General Hospital. For each of these patients, they had whole exome sequencing performed on the pretreatment bone marrow aspirates, and they also performed single-cell RNA-seq and cytoph analysis on the bone marrow mononuclear cell fraction. They also purified CD34 positive cells so that they could enrich this population to look at it in more detail. So single cell RNA sequencing is a very potent new methodology. It's not so new anymore, but it's being more and more widely used, in which you can basically look at the gene expression profile in individual cells in a population. And it gives you the ability, as shown here, to identify different populations within the, with different subpopulations within the initial population. This group out here is representing the stem cell population, and then other populations represent different other more differentiated populations within the bone marrow. So they applied machine learning approaches to identify changes in these populations in the different groups that might have a predictive power, changes in these populations which may predict which patients might belong to group A, B, or C. And what they found out here was that the groups that were most informative in terms of classification were, one, the hematopoietic stem cell population, and then secondly, the NK cell population, and finally, the plasmacytic dendritic cell population. And in terms of the changes that they saw, they found that the patients in group A, which were the patients who responded well, showed an erythroid bias in their leukemia stem cells. That means that these cells showed increased expression of erythroid regulating genes and erythroid regulating transcription factors. And functionally, they showed that these cells were prone to develop more into the erythroid lineage. And they further confirmed that these leukemia stem cells were extremely sensitive to imatinib treatment. The second factor that they found, which was very important in predicting, was the changes in NK cells. And this fits in with work that's been done by other groups, including Sadhu Masjoki and others, who have shown that the presence of NK cells may be predictive of response in CML. And what they found was a large expansion of a particular kind of NK cells called adaptive NK cells, and this reflected an activated state of NK cells. And in the group that did not respond, if they looked at group C, what they found was that instead of erythroid bias, they found stem cells that showed an inflammatory phenotype. They showed increased MYC and IRF1 transcription factor driven genes. They also showed that their NK cells were expressed NKG2A, which is a marker of NK cells being in an inhibited state. So if you look at it, the group A, which is the good group, had erythroid lineage skewed hematopoiesis and activated NK cells, whereas group C, which was the group that failed, had this inflammatory leukemia stem cells, and they had an inhibited state of NK cells. So basically, they took these features and, through their statistical analyses, they identified 8 features, which are along the lines of what I've already talked about, which was significantly correlated with imatinib sensitivity or resistance. For example, erythroid lineage skewing and erythroid program in leukemia stem cells, adaptive NK abundance, NKG2 positive NK cells, a MYC program in leukemia stem cells, and an interferon gamma gene expression signature in hematopoietic stem cells. And using an unbiased machine learning, they could identify these profiles were capable of predicting imatinib response with greater than 80% accuracy. And they're now working on applying these to clinical biomarker development. And so they've validated the single cell RNA sequencing findings using a custom antibody panel that can be analyzed using CyTOF or conventional flow cytometry. So this is really, so just to summarize, I think what we have here is a very interesting finding. One, which may be of clinical importance in, providing additional biomarkers of response for the future. And secondly, it's also giving us important knowledge about the biology of response, and both in terms of changes intrinsically in the stem cell population, as well as extrinsic changes related to intrinsic changes in the stem cell population, as well as extrinsic changes in the NK cells, which are predictive of initial response and actually long-term response to TKI treatment. I'll also mention that this group is in the process of also analyzing features which are predictive of treatment-free remission. And that was presented, preliminary data for that was presented at the ESH meeting. It was not presented at ASH, but we're looking forward to seeing those findings again in future. So now I'm going to move on to a couple of studies which are related to finding new ways to target leukemia stem cells. And so, as you know, leukemia stem cells are resistant to being eliminated by TKI treatment, and there's a lot of interest in understanding why they persist and how we can eliminate them. And there have been, over the years, many studies which have identified different pathways. And so one of the questions is, which of these pathways is really important? And so the group of Satu Muschoki, as presented by Sheri Awad, really looked at this in an unbiased way by doing a high-throughput drug profiling and CRISPR screening to look for novel vulnerabilities in leukemia stem cells. So they did drug screening, in which, the challenges of drug screening in CML stem cells is the low number of stem cells that are present. And so there's also the issue of commonality of pathways between the leukemia stem cells and normal stem cells. And so their approach was to take CD34 cells from CML patients and from healthy controls. And they did drug screening with 80 drugs at 5 different concentrations. And they obtained the dose response curves. And the drugs that they screened included BCR-ABL inhibitors, PI3 kinase mTOR inhibitors, epigenetic modulators, and a variety of other kinase inhibitors, apoptotic modulators, inflammatory inhibitors, and PPAR gamma agonists. And as you can see, they've chosen drugs which are, amenable to clinical application. And so they tested the in vivo, ex vivo drug sensitivity to 80 targeted drugs. They also looked at 18 drug combinations in CD34 positive cells. They further confirmed their findings using CD34 positive, CD38 negative stem cell populations. What they found was that CML stem cells, in addition to being sensitive to BCR-ABL inhibitors as expected, were sensitive to inhibition of VEGF receptor, to CDK2 and V1 inhibitors, to MDM2 inhibitors, and PI3 kinase and mTOR inhibitors. And when they looked in combination, they found that the combination of imatinib with idasanutlin, which is an MDM2 inhibitor, venetoclax, which is an apoptosis inducer, AZD1775, a V inhibitor, and dexamethasone, which would be anti-inflammatory, led to synergistic inhibition of CML CD34 positive cells. And these findings were confirmed in 34 positive, 38 negative cells. And then, this allows us to consider further follow-up of studies of these drugs in alone and in combination in CML patients. They also did CRISPR screening. So CRISPR is a method in which you can knock out individual genes by specific targeting using guide RNAs. And they used a genome scale CRISPR library to see which genes may be contributing to response or resistance to the selective drugs. So if they enhance the sensitivity, you would see knockout of that particular CRISPR, and if it conferred resistance, you would see accumulation of that particular CRISPR guide RNA. And what they found was that the BCR-able TKIs, imatinib and dasatinib and tivozanib, VEGF TKI, showed a similar profile of resistance and sensitizing genes. And they identified KCTD5 as the top resistance gene to all the tested TKIs. So what is KCTD5? It's a potassium channel tetramerization domain containing 5 gene. It is predicted to be involved in the ubiquitin-dependent proteasome-mediated protein degradation. So basically, it's involved in tagging proteins with ubiquitin and targeting them to the proteasome to mediate their degradation. It is significantly downregulated in CML compared to healthy CD34 positive cells. They further showed that it directly interacts with BCR-ABL and mediates its ubiquitination, targeting BCR-ABL for degradation. And that knockout of this led to decreased ubiquitination of BCR-ABL, and which allowed BCR-ABL to persist and led to imatinib resistance in CML cell lines and in primary CML CD34 positive cells. So the combination of TKI with the deubiquitinase inhibitor enhanced TKI sensitivity in these knockout cells. So this is not something which is, this is not a gene that is immediately druggable, but it is telling us about an important pathway, and it's also telling us about the importance of trying to target BCR-ABL degradation as a potential mechanism to enhance responses to TKI in CML. And this is relevant to the next abstract that I'm going to present, which is an abstract from Michael Deininger's group presented by Milad Romagatham. And this is LPA81, discovery of an exceptionally potent PROTAC degrading native and mutant BCR-ABL oncoprotein in CML. So in this, just as an introduction, the rationale for looking at PROTACs, which are drugs which target proteins for degradation, is that with BCR-ABL, we know about BCR-ABL kinase activity, and that phosphorylates different targets which are important for BCR-ABL signaling. However, there is also known to be kinase-independent function of BCR-ABL, so that even if you inhibit the kinase, BCR-ABL can still bind to different proteins, and the binding of BCR-ABL to these proteins may lead to changes in development of signaling complexes and changes in cell function. So if we want to inhibit the BCR-ABL scaffold function, one can argue that you either need to inhibit those bindings or you need to eliminate the BCR-ABL protein. And so PROTACs are also, which stands for proteolysis targeting chimeras, are molecules that target protein degradation by the proteasomal machinery. And so basically they have two portions to it. One portion which, as shown here, would bind the protein of interest, and another portion which would target the degradation machinery and basically bring the protein to the degradation machinery so that it is selectively degraded. And so BCR-ABL PROTACs have been an area of interest, but so far they have not been terribly potent. And it's also uncertain whether the current BCR-ABL PROTACs are able to degrade BCR-ABL compound mutants. And I mentioned that because that's another potential application of PROTACs, because as we know, these compound mutants tend to be hard to inhibit even with the most potent tyrosine kinase inhibitors. So what Dr. Deininger's group did was to synthesize a library of asciminib-based PROTACs, and he synthesized 52 such PROTACs consisting of asciminib connected via linker to a E3 ubiquitin ligase ligand. And the most potent of these was LPA81, and this degraded more than 90% of BCR-ABL within 12 hours at less than micromolar concentrations. And it showed a high degree of efficacy within cells and was potent against BCR-ABL single mutants as well as compound mutants. And it was cytotoxic in cell models with IC50 values less than 1 micromolar. It also did not show any toxicity against wild-type BAF3 cells at up to 10 micromolar, confirming selectivity. And this group is now evaluating the efficacy of these compounds against primary CML cells, including stem cells. And so as this goes forward, this may be a new and novel approach, to targeting BCR-ABL and potentially, the cells which persist after TKI treatment. Finally, I'm going to talk about a couple of studies, including one from our group, which looked at metabolic adaptation to tyrosine kinase inhibition in CML. So Xiaowei Chu from my group presented this abstract. Basically what he found was that there's a fair amount of, the reason that this was of interest is that groups including Wigner Helgeson have shown that CML stem cells show altered metabolism. They show increased dependence on mitochondria, increased mitochondrial activity, and this activity appears to be important for their maintenance. What was not known was the effect of TKI treatment on CML metabolism. And what he found here was that when CML cells were treated with the TKI, there was an initial inhibition of metabolism, both OXPHOS and glycolysis in CML progenitors. However, if these cells continued to be treated over a period of time, these activities were restored. And we further showed using single-cell RNA-seq that the stem cell populations which were persisting after TKI treatment were characterized by reduced expression of metabolic genes and of MYC gene signatures. And they also had increased stem cell and quiescent signatures. And so these were present prior to treatment and were enriched after TKI treatment. TKI treatment initially led to further broad inhibition of metabolic signatures in quiescent HSCs, but that these were subsequently restored with continued treatment. So further studies focused on transcription factors called hypoxia-inducible factors. HIFs are transcription factors that regulate stem cell quiescence, metabolism, and angiogenesis. And what was found in the single-cell analysis and confirmed here by flow was that TKI treatment leads to increased expression of HIF1 alpha in CML stem cells, and that it also leads to increased expression of HIF1 target genes, and that CML stem cells were sensitive to HIF1 inhibition, and this depleted both murine and human CML stem cells, suggesting that HIF1 alpha might be an important regulatory mechanism in CML stem cells that allowed them to persist after TKI treatment and that could be targetable to enhance elimination of stem cells. There was another abstract from Dr. Guo Chu from Guangzhou in China, which looked at another gene which is very similar to HIF1, which is the gene that we studied. This is HIF2. They bind to similar domains, but they have slightly different downstream effects. But what he found was that HIF2 alpha was also overexpressed in CML bone marrow, that CML onset was delayed in HIF2 knockout mice, and LSCs were reduced, and that TKI treatment combined with HIF2 knockout improved TKI efficacy and extended mouse survival. And this was associated with changes in metabolism with reduced lipolysis and increased OXPHOS. And they also showed that HIF2 alpha was also involved in bone marrow vascular remodeling after the onset of CML. So I think these two abstracts point to a potential area of regulation in CML, which may be important in terms of determining TKI resistance, and for which there is potential to develop targeted treatment in future. So with that, that's the end of my presentation, and I'm very happy to take any questions.
Ravi, thank you so much. There's a really good comprehensive overview of what was presented in the abstracts at ASH. So question from Professor Hughes. So looking at all the new discoveries and what you've presented, which of these do you think is most likely to have a clinical impact within the next 5 to 10 years?
That's a great question. I would say that the work from Singapore, from Tiong Ong's lab, is something which if it is it needs to undergo validation and needs to be tested prospectively. But I think that work in terms of developing biomarkers for CML response at the outset, I think is very compelling. And I think obviously this has been shown so far in one set of samples, but if that work could be validated in an independent cohort, then this would be something that potentially could be applied as an additional clinical biomarker in addition to the biomarkers that we currently use. We classify based on the so-called score and the Hasford score, and now there's increased interest in looking at the impact of mutation seen early at the time of diagnosis. But this approach would be something that could be applied and may be helpful in prognostication and in treatment decision making in the future.
Thank you for the nice overview, Ravi. And one question regarding the Sarju-Mustoki data. As everything was in parallel at ASH, so we were not able to participate in the biology session. And were there, from these drug monitoring stories, were there any clinical data so far? Probably I missed it in your talk. From this drug testing sensitivity,
right,
where all the other drugs came up, were there any data on cell lines or something like that?
So from the drug studies, from the drug testing studies, it hasn't been clinically applied so far. So so far the data that is there is identifying potential targets, and it was performed. So the screen was actually performed using CD34 positive cells. So it's using a relevant cell population, and it was validated in CD34 positive, 38 negative cells, a more primitive population. So I think I didn't see that it had gone on to being tested, using animal models or long-term culture, but I think the important thing is that the drugs that were tested are, I think drugs which are approved and which could be brought into clinical use or into clinical testing, relatively quickly.
So what are some of the treatments which we may be available in the next 1 to 3 years with regards to CRISPR and other TKIs? This is especially with regards to patients with resistance to all TKIs due to driver mutations.
So I think that's a really that's a good question. I think the PROTAC that is being developed by Dr. Deininger's lab is obviously very exciting. I'm not sure, at this point where they're at in terms of taking it into clinical development, but I'm sure that they are very excited about it and they're working on doing that. So in terms of the time frame, I 1 to 3 years is probably optimistic given how drug development works, but I think that is definitely something to look at in the future. There may be, so I think I'm just wondering if there would be other ways to, help, target BCR-ABL for activation using, in terms of driving the protein towards degradation, but I'm not aware of any sort of approach which would be immediately available.
A question on persistence. So is there a common mechanism of stem cell persistence, or are there multiple mechanisms involved in different patients? What's your perspective on that?
My thought is that there are probably multiple mechanisms that are involved. We know that from studies in which we've shown different approaches to target stem cells. Whether these would one would hope that these lead to a common downstream pathway, and what we know from studies is that often we end up looking downstream at pathways involving a transcriptional network of MYC, p53, and other transcription factors. But there seem to be many different ways in which the cells feed into this sort of common mechanism. And I think it's very hard to target the core transcriptional machinery without having a lot of toxicity, to normal cells, and to be able to do it specifically. But I think if we can, so I think targeting the specific mechanisms that lead into these pathways is likely to be more feasible. And this is where I feel that we may have to develop some methodologies in which we can understand the common mechanisms that lead into this, and be able to screen for these, and then be able to use a more personalized, say, approach towards eliminating stem cells in this setting. I think the other thing that was that's interesting that comes from I think as we're learning a lot from the clinical data is that as we look at recurrences, after TKI discontinuation, which is, probably the best measure we have for persistent stem cells. And the data that Suzanne had presented at ESH, showing just showing the and I think which Tim and Susan had also presented, seems to indicate that the patients who respond quickly after TKI discontinuation tend to have a different pattern of recurrence as opposed to those who present late after TKI discontinuation. And so again, if you think about those, it may also tell us that there may be different populations or different mechanisms involved with the persistence and recurrence in those situations. So I think there's still a lot more that we need to learn, but I think it is going to be complex and is going to require a more personalized approach.
Another question come through on the chat. Is there any data on immunotherapeutic approaches for CML? So this is with regards to the recent developments in ALL treatment using the CAR-T therapy.
There are studies that are testing immunotherapeutic approaches. I think and at different levels of specificity. I think the German group has been pursuing the use of interferon, and I think the data related to NK cells, which was which has been there for a while and which was sort of seen in the data from Singapore, is very exciting, and seems to suggest that this is a population that may be particularly active in CML. And so I'm sure it'll generate further interest in methods to enhance NK cell activity, or even adoptive transfer of NK cells in the future. In terms of antibody or CAR T cell treatment, I think the problem with for CML and other myeloid malignancies is the toxicity to normal hematopoiesis as well, which is different from what we see in the B cell malignancies, where it is possible to target B cells without and the toxicity is manageable. So I mean, there's been a lot of interest in finding different cell surface markers which are characteristic of CML stem cells. I see Peter Valence on the call, and he's done a lot of work related to that. I'm not aware of studies actually using that to target stem cells. Thor Fiorettos had many years back described IL-1RAP as being a potential marker, and that I believe that is under development as a immunotherapeutic, but it's being tested right now mostly in solid tumors and not in leukemia.
Thank you. Are there any other questions? If not, perhaps I will ask one that I would have really liked to ask both you and Mike, both your opinions. So to finish off, where, in your opinion, are we able to predict whether patients will be able to remain in TFR after stopping TKIs?
I think we made progress, but I think we, in short, I think we made progress, but I don't think we're quite there yet. I think we, I think we need, we made a lot of progress in terms of the clinical markers, and I think work done, by the group at Adelaide and Suzanne and others in the TFR studies has clarified, better from the clinical point of view as to who is more likely to, maintain TFR versus not. But, I would argue that we still don't have a very good idea as to why some patients maintain TFR and others don't. What is the real biology of it? And so I think that studies along the lines of what the group at Singapore has done, but maybe, I think their studies were done at disease onset. Maybe a similar study, done at the time of treatment discontinuation may be helpful. I think most of our biomarkers now are done, in blood. It's relatively hard to get bone marrow samples from patients, at that time. And that may be important in terms of, understanding, the biology of this. On the other hand, in terms of a biomarker and prediction, it's obviously much more convenient if we can do it on a blood sample. So I would say that, we have some clues. We sort of know that there are characteristics of the stem cells that are associated with response. We know that there are characteristics in the environment which are associated with response, but to actually have a good idea to be able to predict who's going to respond or not is, I think, we're still lacking.
Yes. Yes.
But
much has been done.
But, I think it's probably the most important area to work on. One of the most important areas to work on, besides the patients who fail all treatments. Yes.
Yes, definitely. Yeah. Well, I guess sort of leading from that, one of the major projects of the iCMLf is the TFR Alliance and working on that. So it'd be interesting to see what data comes out of our partnership with the Harmony Alliance with the big data analysis sort of within 2023. Hopefully towards the end of next year, we'll start to see as well. Ravi, I would just like to thank you so much for being so generous with your time and taking more time to perhaps fill in when Mike wasn't available. We really appreciate your efforts on answering all the questions. For our audience, please join me in thanking Professor Bhatia for his time. And also, if anybody in the audience has any suggestions for future topics for next year that we can discuss and invite experts to share their opinions, we'd be more than happy to hear your ideas and opinions. So I will conclude by saying I wish you a very Merry Christmas to everybody who celebrates and a very happy holidays to you all.